Good morning. It's August 1st, and this is your daily brief in science. Here's everything you need to know. Recent studies suggest a troubling link between respiratory viral infections and breast cancer. Research indicates that infections like influenza and COVID-19 may awaken dormant breast cancer cells in the lungs, potentially increasing the risk of metastasis and mortality. Led by James DeGregori from the University of Colorado, an international team conducted experiments on mice to explore this connection. Their findings highlight the role of the immune molecule interleukin-6, or IL-6, which appears to activate these dormant cancer cells. Notably, mice lacking IL-6 did not experience the same proliferation of cancer cells. While experts recognize the significance of these findings, including Professor Dr. Andreas Bergthaler, they stress the need for further research to determine their relevance in humans. The implications for different cancer types and subtypes of breast cancer remain uncertain, as the current results are preliminary. DeGregori emphasized the importance of treatment balance, warning that certain anti-inflammatory drugs could weaken the immune system, complicating care for cancer patients facing infections. Future research aims to delve deeper into the mechanisms behind the reawakening of dormant cancer cells and assess the impact of vaccination on this risk. Existing COVID-19 treatments targeting IL-6R and JAK1/2 inhibitors may offer potential solutions to mitigate infection-induced metastatic cancer progression, though more studies are needed. After recovering from viral infections, the study found that cancer cells stopped proliferating but showed changes in gene expression linked to tumor metastasis, indicating a long-term risk for recurrence. Critics have raised concerns about methodological flaws in the study, including possible misclassification of causes of death and statistical biases, highlighting the necessity for more robust analyses. This research, published in the journal Nature, involved collaboration among institutions from the USA, UK, and the Netherlands. Recent research has shed light on the origins of potatoes, revealing that they evolved from a tomato ancestor nearly 9 million years ago through a process of hybridization with a plant known as tuberosum. This significant hybridization event led to the formation of the potato lineage, which includes the cultivated potato, Solanum tuberosum, along with 180 wild species. The study highlights how hybridization can give rise to new plant lineages, exemplified by the development of tubers that store nutrients. Notably, while interspecies hybridization often results in sterile offspring, this case produced a successful organism capable of thriving in diverse environments. Potatoes adapted remarkably to high-altitude Andean habitats, leading to a vast diversity cultivated by indigenous peoples, who developed hundreds of potato varieties. Today, potatoes are recognized as the world's third most important staple crop, providing 80% of human caloric intake alongside maize, rice, and wheat. Their versatility and low greenhouse gas emissions contribute to their status as a climate-friendly food, making them vital for sustainable agriculture. Introduced to Europe in the 16th century, potatoes initially faced skepticism, but eventually became a nutritional staple due to their adaptability. Published in the journal Cell, the study emphasizes the importance of hybridization in evolutionary biology and its implications for agriculture. While hybridization can yield negative genetic outcomes, it can also produce beneficial combinations, as seen with the 107 wild potato species that exist today. This evolutionary process occurred long before the common ancestor of humans and chimpanzees existed, underscoring the deep historical roots of these plants. The findings open avenues for developing more resilient crops with enhanced nutritional qualities, aiming to improve biodiversity and agricultural sustainability. A recent study reveals concerning levels of microplastics found in indoor air, particularly in homes and vehicles. Conducted by Nadia Yakovenko and her team at the Université de Toulouse, the research indicates that the concentration of microplastics, especially particles measuring between 1 and 10 micrometers, is significantly higher than earlier estimates that primarily focused on larger particles. Over 90% of the identified microplastic particles are under 10 micrometers in diameter, raising alarms as these tiny particles can penetrate deep into lung tissue and potentially enter the bloodstream. The study highlights that vehicles have particularly high concentrations of microplastics attributed to confined spaces, heat, and the prevalence of synthetic materials which release microplastics through friction. This increased exposure risk in cars is particularly troubling for commuters, including children and individuals with respiratory conditions. Despite these findings, environmental health scientist Professor Kevin Thomas warns that the study's limited sample size may not accurately reflect real-world exposure levels. The researchers did not measure microplastics directly in the human body. Instead, they estimated potential inhalation based on air sample analyses, introducing uncertainties into their conclusions. The calculations rely on air samples and standard breathing volumes, highlighting the necessity for further extensive research to refine these estimates. To reduce exposure to microplastics, individuals are advised to limit single-use plastics, avoid heating food in plastic containers, and enhance indoor air quality through ventilation and filtration systems. Practical measures include regularly ventilating indoor spaces and using HEPA filter vacuums or air purifiers. In response to these findings, an editorial from the World Health Organization has called for an international plastics treaty aimed at capping plastic production and addressing health implications. The study's results were published in the journal PLOS1, underscoring the urgent need for more comprehensive studies to fully understand the health impacts of microplastics.
Researchers at IIT Madras have made significant strides in the fight against plastic pollution with the development of a biodegradable packaging material derived from agricultural waste. This innovative project leverages fungi, specifically Ganoderma lucidum and Pleurotus austriatus, to promote a waste-to-value approach that diverts agricultural residues from burning, aligning with circular economy principles. Laboratory studies have demonstrated the feasibility of these mycelium composites, showcasing promising mechanical properties, water resistance, and biodegradability. Current efforts are focused on optimizing these materials for market scalability. Notably, Ganoderma cultivated on cardboard has exhibited exceptional compressive strength, surpassing traditional plastic foams like expanded polystyrene, marking it as a viable alternative in the packaging sector. To bring these biocomposite products to market, a startup named NatureWorks Technologies has been established, co-founded by lead researcher Dr. Lakshmanath Kundanati. The startup aims to facilitate technology transfer with industry partners, addressing environmental concerns while creating economic opportunities for rural communities by valorizing farm waste. Research is also underway to integrate various fungal strains and substrates to enhance the properties of these biocomposites, potentially adding thermal and acoustic insulation capabilities. The mycelium-based material is tear-resistant, does not require harmful chemicals for processing, and is safe for consumption, indicating its versatility beyond packaging. In addition to packaging, researchers are exploring the use of this fungal paper for compact biodegradable batteries, reflecting a push towards sustainable electronics. The mycelium's properties may also serve as an excellent emulsifier, which could benefit the production of plant-based cosmetics and foods. This development is a crucial step in mitigating plastic pollution, which poses significant threats to wildlife and ecosystems. The findings of this research were published in the journal Bioresource Technology Reports, highlighting the environmental benefits of these new materials. Researchers have made a groundbreaking discovery by digitally reconstructing the tattoos of a 2,500-year-old Pazyryk mummy, providing new insights into ancient tattooing practices in Iron Age Siberia. This mummy, found in the Altai Mountains, belonged to the nomadic Pazyryk culture, which buried its dead with intricate artifacts, including horses and ornate rugs. Initial examinations of the mummy revealed that the tattoos were obscured due to darkening from the mummification process, prompting researchers to employ advanced imaging techniques to uncover them. The findings, published on July 31st in the journal Antiquity, represent a significant advancement in understanding ancient body modification practices and the social context surrounding them. The analysis revealed numerous tattoos on the mummy's hands and forearms, featuring motifs such as birds and complex hunting scenes, created using puncture techniques akin to modern hand-poking. Interestingly, the tattoos on the right forearm were found to be more detailed than those on the left, suggesting the involvement of different artists or varying skill levels over time. This study indicates that multiple tattoo artists may have contributed to the designs, employing advanced techniques that go beyond previous hand-drawn interpretations. Dr. Gino Caspari, the senior author of the study, noted that tattooing should be regarded not merely as decoration, but as a skilled craft requiring technical ability and training. Furthermore, the tattooing process may have had ritual significance, highlighting a complex relationship between tattoos and identity within the Pazyryk culture. Notably, post-mortem burial incisions were found to have cut through the tattoos without any effort to preserve them, suggesting that these markings were significant primarily to the living. Experts stress the importance of these ancient tattoos as reflections of individual lives and cultural beliefs, expressing hope for further investigations using advanced imaging techniques on other preserved tattoos. The urgency for digital preservation is underscored by the threat of climate change to the frozen tombs that have preserved these remains for centuries. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.